So tonight we have a variety of speakers who are going to come up and talk about the two programs that you're here to listen about. And we also have some people watching online, so I just wanted to welcome them also to the info session here. So just an agenda for the evening tonight. So my name is Erin Friesen, and I'm the Acting Associate Dean for the Lab and Allied Health portfolio. So clinical genetics and medical laboratory sciences are two of the programs in this portfolio. If at the info session you want to talk to me about some other programs that I'm also responsible for are prosthetics and orthotics, biotechnology, occupational health and safety, food science, and environmental health. But we have some presentations here, so we'll have the two program heads from the different areas come and talk. So we have Irene and we also have Andre who are going to come talk about their program areas and give you more information. We also have some graduates of the program who are going to come talk. So we have Phil, who is a graduate from the clinical genetics program, and he's going to talk about his experience in the program and where he ended up. And then we also have Edward, who's a graduate of the program, and he's also going to talk about the program and his experience. And a lot of you may have questions about whether or not you have the entry requirements to get into the program. So we also have Gabriella from Program Advising, who's going to talk about the process of applying for these programs. Because we know a lot of you may have questions and they might be specific to your own case, what we're going to do after the info session is we have a room just booked right behind here, and we're going to do an info session where you can go and talk to, if you're interested in clinical genetics, they can answer your questions. If you want to talk to the students, they can answer your questions. Or if it's program advising questions you have, you can also speak to them about your particular questions. So why choose BCIT? BCIT is actually a really exciting place to be. I'm a graduate of other schools, but I'm actually really happy to be working at BCIT because I'm very proud of the type of education that we deliver here. One of the things that's really great that I get to talk about is how graduates are job ready at the end. And so just to give you some numbers from the medical laboratory program, this is the numbers from the 2017 survey, so they would be graduates from a year or two before that. 98% of medical laboratory sciences grads are working in the field after graduation, and all of them found employment within 6 months. So it's quite high, the rate of success. One of the other exciting things about BCIT that's different from universities is you work in a cohort model. So you will be paired with groups of ranging between 8 to 20 students, and you will participate in your labs with a very small number of students, and you'll carry through your whole education with this small group of students. So you get to know your peers quite well, and then in these small groups your instructors get to know you quite well as well. It's a very supportive learning environment, so the instructors who are in these programs have extensive education, and they're also there to support you, whether it's office hours. BCIT also has a lot of great support, whether it's extra English support you need, or if you have a disability that needs some special consideration for your education, the accessibility department here can help you with that. There's also a gym, so we encourage students to be active. There's lots of variety of different things that we can help you with. The faculty who is hired is very clinically relevant. They've all worked in the field, so when they're teaching you, they're able to draw upon their own experience in the education. The two programs we're going to be talking about tonight are accredited programs, which means we're teaching to national education model, and they're nationally certified. So what you are learning would be similar to programs delivering the same program across Canada, so you can move to other areas of the country if you were wanting to work in other areas. So one of the things you'll want to think about before you come here is whether or not these programs are for you. So they are lab programs, so you would be working in a lab. You need to be self-sufficient, so at times you might be working independent. At times there's going to be time pressure, so you need to be able to work under pressure. These are science programs, so you should be curious and fascinated by science. process orientated and accurate. You don't want to mess up somebody's label on a lab sample, as there's implications for that for the patients. You need to be able to work independently as well as part of a team. So attention to detail is quite important. Manual dexterity, you will be working with your hands, you'll be working in a lab, so you should be able to do things like pipetting. You need to have keen observation skills. You will be doing a lot of lab work, so following strict procedures. And you need to have a high level of physical and mental resiliency. So a student here at BCIT, what makes BCIT different is it's quite an intensive program. So you will be in class for long hours. Often that might be from 8:30 to 4:30, 5 days a week. We do give students a break on Wednesday afternoon where we encourage you to get involved with BCIT activities, whether that's the student association or time to go to the gym. But it is very time intensive, where you'll be in a combination of both labs and lectures. BCIT's programs here are a lot different from other programs, where we actually have a lot of time spent in the lab where you're getting industry relevant experience. You'll be using quite high tech equipment here, so we like to make sure that what you're learning here is reflective of what's also in industry. So it's full days, Monday to Friday, small class sizes with direct contact with your instructors, and hands-on experience. So you'll be doing cell culturing, viewing chromosomes under microscopes, analyzing blood samples. So there's a variety of things that you'll be doing in these program areas. So the two programs we're going to be discussing today are very similar programs in the sense that you will be employed either in hospital or clinical settings, but the actual content you'll be learning in these two programs are slightly different. Another small distinction between the two programs is also clinical genetics requires you to have an undergrad degree. So it is a post-undergrad degree, and it's more specialized in a more smaller program. The medical laboratory science program, on the other hand, you can enter after high school, but it is a competitive entry program, and Andre will speak a little bit more about the entry process into medical laboratories. Medical laboratories, you learn a much more diverse variety of programs, so you might be learning about microbiology or transfusion or chemistry, whereas clinical genetics is much more specialized in that one field. So with that, I'm going to pass the floor on to our speakers. Just to remind you, questions, if you have questions, we're going to allow those to happen in Town Square A, which is right behind us, so we'll be able to more directly answer those questions. So with that said, our first speaker is Irene Dorocic, who is the program head for clinical genetics.
Thank you very much for that introduction, Erin. And welcome everyone to our info session. We're so glad you took the time to be with us either online or in person here at Burnaby. I am again the program head for the Clinical Genetics Technology program, and I wanted to give you some information that can be a useful supplement to what you can find at our program webpage. As you can see in the picture here, this is a lab-based profession. So while you will be an important part of healthcare, you will not have direct patient interaction. Instead, you'll be spending long hours in the lab working with chemicals and potentially biohazardous specimens from patients, such as peripheral blood or samples from women that are pregnant. So these are some things you need to keep in mind when you are considering this career. So this program is one of only 2 programs in the country. We are a very specialized program compared to the medical laboratory program, and our focus is on genetic disease. And in teaching the theory, the lab-based procedures, and the professional attitudes that we would like to see for people in this profession. As you can see on this slide here, we focus on 4 different areas for the assessment of genetic disease, as we are trying to answer questions such as, is there a chance I can have a child with a genetic disease? Do I have a genetic disease? Is my genetic disease in remission? These are all questions that our technologists create the data for, and most importantly, also analyze the results that they have in the lab. After graduating from our 13.5-month program, you are eligible to work at the entry level anywhere across Canada in a clinical genetics lab, which tend to be in the hospital environment. We also allow you to write the national certification exam, which is needed for employment in this field. As you can see, we have 4 different technologies in our program, starting with classic cytogenetics, which is the assessment of chromosomes for structure and numerical abnormalities. So you'll be doing procedures like working with patient blood, culturing, light microscopy, preparation of chromosomes for analysis, and we will show you how to use the specialty computer programs that are needed for analysis of this data. We also do work directly with the molecules of DNA and RNA, which are genetic materials. So here you might be isolating DNA from a variety of samples, performing procedures like the polymerase chain reaction, Sanger sequencing. We have started to introduce next generation sequencing as well. The last 2 technologies we have are a combination of classic and DNA-based procedures. With FISH, we have fluorescently labeled DNA that is used to assess chromosome abnormalities too small to be seen by classic cytogenetics. And then finally, chromosome microarray is a slide-based technique. Once again, you are looking for chromosome abnormalities. We are here to try to answer the patient's questions regarding their health for genetic disease, which are diseases such as cancer or Down syndrome. So again, here we have a picture of people working in the lab. Important things I'd like you to notice is we have some microscopy happening. Safety is very important as these people are properly gowned, so we will show you the proper safety techniques to use. The lab environment here at BCIT is a simulation of the hospital environment because we feel this is the best way to prepare you for your time in the hospital. And again, we also have some computer analysis of data as well. So since you're here to know about how to enter the program, let me speak a bit about our entrance requirements. As Erin mentioned in her introduction, this is a post-degree program, which means you must have graduated from a 4-year Bachelor of Science or a higher degree. We will also consider people who are in the 4th year of their bachelor's program if they are scheduled to be complete before the program begins in September. Notice that we're looking for people in courses like cell biology, genetics, molecular biology. The higher your marks, the better. We also are looking for graduation within the last 5 years. Be aware as well, you need to have documented evidence of your English. We're looking at the equivalent of a grade B in English 12. So you'll either support this with your high school transcript, your university transcript, or you'll need a English assessment test like TOEFL or IELTS. The program advising people can give you more information about how to get these tests done. Our program, as I say, is 13.5 months. So we start in September and then are complete by the following October. You will spend 25 weeks here at BCIT, beginning in September and continuing till the middle of March. Once you are done with this, as it mentions here, there is a 30-week clinical practicum, which we think is an excellent and important part of your training, as you will actually be in a real hospital lab working with real samples, surrounded by graduates of our program. Notice, however, some of these placements are outside of the Lower Mainland. We always make it very clear to applicants that we distribute these practicum at random. And if you have one of the practicum from outside the province, from outside Vancouver rather, you will need to be responsible for making those arrangements and dealing with those costs yourself. So I've been saying a lot here as the instructor of this program, but we believe very useful information can come from our program graduates. So we're very fortunate that my recent graduate, Phil, is here to tell you about his time in the program. So I'll let Phil talk.
Hey guys, you might have to put up with my voice a little bit. I'm fighting a bit of a cold today. So my name is Philip. I am a recent graduate in October. I've been working for about 3 months now at BC Cancer Agency. I was lucky enough to find a job about 2 or 3 days after I graduated. So a little background to myself. I am a graduate of SFU in the Bachelor of Science department there in Health Science. About 2 years before I graduated there, I decided to go to an info sesh like this one for both med lab and clinical genetics, and I discovered the clinical genetics program at BCIT. And from then on, I sort of started to tailor the courses and my co-op programs at SFU towards getting into the clinical genetic technology program here. And that's one thing I can't stress enough. If you're still in university at this time and looking to get into any of these programs, I would highly recommend going and doing co-op while you're still at university. It helps to take a co-op that specializes in some sort of lab skills, because from then on, you're able to actually build your resume to get into this program as well. So after I graduated, I took about a month to work through the application program to get into clinical genetics, because it's a pretty long one. And so once I got in, you start to see the positives of the program. Because I didn't want to do a 4 to 5 year PhD program at SFU, I gravitated towards the 13-month program here at Clinical Genetic Technology. They break it up into a 6-month study program where there's a lot of application. And after the 6 months, you are then sent to, at least in my case, I was sent to BC Cancer Agency for about 3.5 months, and also Royal Columbian Hospital for 3.5 months to learn all the different forms of technology in this field. The nice part is also about being in this program is all your professors also work out in the field. So right now I work at BC Cancer Agency. 2 of the professors that I had while I was at BCIT are now my colleagues also at BC Cancer Agency, along with the practicum site at RCH. One of my professors was also a colleague there as well. So you're able to build a nice network of people through your professors, and because it's such a small, tight-knit community in BC, from what I know, there's only about 4 or 5 sites that have genetic labs. You get to know everybody pretty fast and well there. The typical questions that people have about this program are, what's the workload like? Can I hold down a part-time job? I would say that the workload in the first semester was not overwhelming at all. As long as you stay on top of your studies, you'll be fine. I was able to hold down a part-time job while I was in the program. The second semester picks up a little bit, and you'll have some tougher days here and there. But for the most part, as long as you stay on top of your studies, everyone is pretty good. As for the field itself, there's high demand at all the hospitals across the country. Most of my classmates, I would say 1, 2 are still in BC. 2 of them are in Edmonton, 1 of them is in Calgary, 1 of them found a job in Hamilton, and I think I might be missing 1 other person, but there's still a lot of other jobs that are able to be found in Vancouver, Calgary, Edmonton, and if you're even looking to go to Ontario, there's jobs there available as well. I can't say enough good things about the program. I got help at all times when I needed it. All your professors are accessible at all times for one-on-one or through email. They answer their emails very quickly. They're all super cool to hang out with as well. I'm now friends with the professors I was with at BC Cancer Agency as I work with them now. And I highly recommend the program to anyone who's interested. Thank you.
All right, next we have Andre, who will be speaking about medical laboratory sciences.
Welcome, everyone, and again, thank you for coming tonight and taking time out of your schedule. I know it's a busy time right now, so we do appreciate it. So I'm going to talk about the MedLab Science program for the next little bit here. We're switching gears. So the medical lab technologist is an important role in the hospital. So as you saw in the video that was circulating at the beginning or looping through, they are an integral part of the hospital. There's a number of different departments that people can work in. And typically what you're going to be doing in the laboratory is you're going to be working with a lot of high-tech equipment and instrumentation. And really, you're going to be assessing and looking at different specimens, so blood, body fluids, tissues. All of that stuff is then going to be processed in the lab. It's going to be analyzed, and it's going to be produced in reports for the physician. And there they're going to be able to make look at it for diagnostic purposes. But more importantly, they're also going to be looking at it for treatment therapies and as well as monitoring people's health. So there's diagnostic principles to it, there's monitoring therapy, and looking at treatment plans. So that's really a lot of the work that we do and what it supports in the hospital and in that system. So as you can see here on the slide, 85% of the decisions made in the hospital about diagnosis and treatment are based on the laboratory results, and that again works with the clinical genetics department as well. So as you can see, there's a lot of synergies between both programs here at BCIT. In the profession, there's typically 6 key disciplines that people can work in. There are other departments as well that people tend to work in, but the 6 that we're going to focus on tonight, I'll show on the next slide. But really, those are the areas that you're going to be trained in as well here at BCIT. Just to give you a little bit of perspective, similar to clinical genetics, it is the format is a lecture similar to this, as well as a laboratory component. So it's a lot of hands-on work in our program. It's a lot of labs. So you're going to be going to lecture and then practicing in the lab. And we do have a really good equipment list and instrumentation, so you'll get a chance to practice with all of mostly relatively new equipment. But you get the sense. So we try to simulate as much as possible in our lab scenarios. So again, you're going to build a really good relationship with your instructors and the other students within your program. Okay, so it is a cohort model. You'll be going together, 80 students, and then we split you up into 4 sets of 20. And from there, you're going to go to your different labs. Just again, from a program perspective, the way it's sequenced is you're going to be starting in next January, and that is your level 1. So a lot of that is your introductory courses, or a lot of the courses that is going to set you up for these 6 disciplines that I'm going to show you. A lot of that work is going to get you ready, and then in the spring, after level 1, you're going to be doing a phlebotomy rotation or a clinical. So you're going to be going out to the hospitals and the private labs to collect blood, and then in the fall, you're going to come back into level 2, and there you're going to be taking on these 6 disciplines. And then you move on to level 3, and then you're going to go into your long block or your big clinical practicum. All right, so that's kind of how the sequence of the program works. So it's 3 levels here at BCIT, and then long block in the next round for your clinical portion. So just to kind of give you an overview, there's six of the disciplines that we focus on here in our program. So you've got clinical chemistry, histology, molecular diagnostics, transfusion science, microbiology, and hematology. So those are the different areas that you could be working in. You're going to get trained in all of those when you go to the clinical environment. So you're going to be working with a preceptor in each of those areas. They're going to show you, you're going to take all the stuff that you've learned here at BCIT and you're going to apply that out in clinical. All right, and then you're going to build on those competencies that is outlined in the CSMLS, or the Canadian Society for MedLab Science profile. So all of those competencies are going to be taught here at BCIT, and you're going to build those skills in order to graduate and then challenge the national exam. So just to give you an example, so my background, I came from Alberta, and then when I moved here, I started working in the clinical chemistry department. So again, in that department, we're looking for things like drugs of abuse, different hormone therapy. We're looking for your general electrolytes, liver profile, kidney, all that kind of stuff. So when you go to the physician, you get your requisition, they're going to draw your blood, and from there, those specimens get sent to the lab. We then process them and then report them out. So from my history, it comes from the clinical chemistry. There are other departments that you're going to get trained in, and then you can work in any of those. So they all are pretty unique in their own ways. They all have different instrumentation. I'm not going to go into each and every single one specifically because they are in itself a fairly extensive unit or department. But just know that they are, again, they have lots to study. So I think this is a good time to just kind of move into sort of the admissions process. So with the admissions, again, it's similar to clinical genetics, but in the sense that, again, you'll have to have strong communication skills and proficiency in English, reading, writing, speaking, etc. So again, that's something, a key requirement for our program. Also, you have to ensure that you meet the equivalency of the grades as well as within recency. Okay, so those are the prerequisites. We have our admissions process is in 3 steps. So the first steps is, did you actually make the grade requirement and the recency? Next step is you're going to go through an MMI process. So that's a multiple mini interview. In that session or during that stage of the admissions process, you're going to be going through a number of stations that's going to look at both cognitive and sort of your social aspects of your learning. And so we're going to sort of assess you based on those things in different little interview style stations. And then from there, we're able to generate a score. And then from that, the top 80 will get in. Okay, so stage 1, meet the grade requirements. Stage 2, get through the MMIs. And then from there, we offer out our applications, so you'll be able to get in. So that pretty much highlights our admissions. I know we have program advising here that's gonna help us go through that a little bit more. So I'll definitely open the floor to some of those questions. But at this point, I think I'll leave it at that. I think, yes. I think that's it. So that's MedLab, the program. So again, the main places that you're going to be working is in the hospitals, private labs. We've even seen people move into technology roles, such as a lot of the instrumentation you're going to be working with. Sometimes those people get hired on to go work at biotech companies. So there is a way to sort of explore the profession a little bit more. So I will leave it at that, and I think our next speaker is our alumni and now instructor. Oh, sorry, no, we're doing clinical placements, my bad. We have to still talk about the clinical. So I've just went over the didactic, and then we've got Debbie who's gonna go over the clinical placements. Thank you.
So again, welcome everybody. My name is Debbie Power. I'm here on behalf of Donna O'Neill, who's the clinical coordinator for the program, but she's away this week, so she asked me to speak on her behalf. As Andre indicated, at the end of your first level, you'll complete a 2-week phlebotomy rotation, and that happens in the end of May, beginning of June. And then at the end of your 3 levels of academic, you'll go out for a 38-week multidiscipline placement. Things that are really important is when you're out in those placements, you can expect to be scheduled in a 24-hour setting. So you could be working 7 days, evening shifts. As well, when you go out to your clinical, you are responsible for all of your associated costs that are incurred, including transportation and accommodations. Our clinical placements are located in accredited labs across the province, so they are in the Kootenays and the Interior and the Lower Mainland and on Vancouver Island. The clinical partners in those areas include Fraser Health Authority, Interior Health, Life Labs, Providence, Provincial Health Services, Vancouver Coastal, and Vancouver Island. The most important thing is, where are you going for your clinical placement? Well, the program uses a computerized program and utilizing an automatic matching system in order to assign those clinical seats. You'll be given an opportunity to enter 5 preferences in, and then the system is run an algorithm in order to make that selection, and it's done randomly. What's really important is that there is no guarantee that you will get one of those 5 clinical placements that you have as a preference, and you have to be prepared to go to any one of our clinical seats located throughout the province, because we have 80 seats and we have 80 students, and so we have to fill all of them. So it's really important to sort of to think about that when you're entering into the program, that you are prepared to go anywhere. Once the site selection has been completed, all of those sites are finalized, and there's no switching and there's no trading. So again, you'll have to be prepared to go wherever your site assignment is within the province. And that's it. I'll be happy to answer any questions in Town Square A at the conclusion of our session here. And I would like to hand it over to our alumni, Edward Zhao.
Hi, so my name is Edward. I'm currently one of the instructors in clinical chemistry for the MedLab Science program. And back in 2010, I enrolled in this program, and I was just like any other science graduate students out there, hoping to get into the healthcare industry and start my career there. But I realized that it was very competitive, and a lot of times you would need to acquire a new diploma at BCIT to be able to work in the healthcare field. So when I did my research, I found that the BCIT MedLab program offers training in 6 different disciplines, which allows you to work inside a hospital setting. And that started my journey and pushed me in the right direction. In my first semester here at MedLab at BCIT, they laid the foundation of clinical testing. So with that, it prepared all the students to head out into clinical rotation after the first semester in their phlebotomy rotation. So we learned how to take blood in the first semester amongst other clinical testing foundations. And after the first semester, I was actually hired on as a lab assistant right away with the private labs at the time, BC Biomedical. So it was a very rewarding experience. Entering into the second semester of the program, the workload became much heavier. So part-time jobs, you would really have to be able to manage your time very well. So that's something that I would advise for all the applicants to this program. Starting in the third semester, you truly learn a deep appreciation for all the testings and all the simulations that we do. And of course, you're looking forward to your clinical rotation at the end of level 3, where you spend 30 weeks out in either the hospitals or the private labs to carry out your practicum in each one of the 6 disciplines. Overall, BCIT MedLab program prepares you very well for your clinical rotation, and the job prospect is excellent because this does span across 6 different disciplines. And actually, there are other lateral disciplines that you could also work in as well. For myself, I did 2 years stint with parasitology with Life Labs, so that's also another option as well. Some of my colleagues have gone into virology at BC Children's Hospital. So in terms of job prospect, there are many opportunities out there. And also you could have the opportunity to work for an instrumentation company, like Andre has mentioned, being a sales rep or a service rep. Also, you could work in the field of quality assurance, either within the health authority or an external accreditation body as well. Also, if you have the love for education and teaching, you can also become an instructor later on as well. So overall, this program has provided lots of learning experience for me. And one of the biggest things, one of the challenges definitely is the workload. And starting in the second semester, you will feel that for sure. But at the same time, we have a very strong support system through our peers as well as our instructors, and we want you to succeed. So we're here for you every step of the way. So you definitely have the resources you need to be successful here in this program. But overall, the MedLab program is excellent. If you're thinking about applying, I would strongly encourage you to do so. Start your career today.
All right, so before we get to the next section, I remembered I was supposed to say something earlier when I was talking about the two programs, which I know is probably a question you guys are maybe thinking in your head at this point. And of course, I want to encourage you to follow your passion, but I know another thing at the back of your mind is, well, how much money am I going to make? So I just wanted to mention the salary from these two programs are very similar. Clinical genetics, so these are recent grads. Clinical genetics had a starting salary of $29 an hour, and medical laboratories was $28 an hour. But before we go on to the next, or I guess we are at the next session, so I'd like to invite Gabriella up to talk about program advising, so she'll talk about some of those details about how to apply for these programs.
Good evening, everybody. My name is Gabriella Nava. I am a program advisor here at BCIT. And so at program advising, we help you along the way with many aspects of the application process. We can help you to understand what are the program entry requirements that you will need in order to apply to the program, as well as understanding how competitive the program is. We will help you along the way if you shall need any upgrading. We can guide you towards where you can do those upgradings if you need them. So we also assist you on how to make your application stronger, because health programs, especially these two that you just heard about, are quite competitive. We also help you with the details that you will need to get into the process of the application and the start dates and the deadlines and all of that. The application process is very important that you will know how you have to follow it. You have to make sure that you review all the program entry requirements. You also have to make sure that you provide the proof to demonstrate that you meet all those program entry requirements. Once you have completed also the mandatory applicant questionnaire and the acknowledgement documents that the programs have, you can now be confident in applying to the program. And it is important for you to apply early so you can be before the deadline in case there is something that you might need before the deadline. We strongly suggest to never leave your application too close to the deadline. Once you have all of the requirements and all the documents ready, then you can submit your application. And at program advising, we can also help you to become familiar of the steps of your application. So what happens after you apply? After your application, after the application deadline, there are certain processes that happen. So the department shortlists the applications, then the mini interview happen, and after that the decision is made. So we would guide you at program advising with all these aspects of the application. Also at program advising, we want to make sure that you are taking advantage of all the services. We have a student association dedicated just for students. They can help you in many aspects to be successful while you are in your studies. They can help you by offering a beautiful library that is open pretty much every day of the year, except for a few days, and it's open 24 hours a day as well. You have access to employment help when you are ready to apply for employment. The student association will help you with that. It is also important that you know that you can exercise here and have fun and go to our fitness facility. It has very modern equipment, so you can have balance in your life while you are studying here at BCIT. There are also health services offered and open for students. That is important for you to know that there's other resources, not just the studying here at BCIT. It is important, but also the lifestyle. There are clubs that you can join and have fun. So at Program Advising, we help you with the program entry requirements, the process, and we make you aware of services that BCIT has for all of you while you are studying here at BCIT. So if you are interested in checking all the programs, such as Spend a Day, we have a great program such as Spend a Day where you can become a student for half of a day and they match you with another student so you can experience what a program is about. We have student blogs. We have a lot of great videos on YouTube. I strongly encourage you to take a look to all the videos that we have on YouTube. And of course, we also have Facebook and we have great pictures if you go to Flickr. If you need to connect with us, we are available in different ways. You can meet with us in person 3 times a week. We have something that we call drop-in sessions. We don't provide appointments, but you can come to these drop-in sessions 3 times a week. Tuesdays and Thursdays are in the afternoon and Wednesdays are in the morning. You can also connect with us via telephone pretty much every day of the week except for Wednesdays. And you can always connect with us through sending us an email at program_advising@bcit.ca. And as my colleagues mentioned, if you have further questions, we will be very happy to help you as much as we can today. If we cannot answer questions today, if you have questions that are more specific, we will invite you to connect with us through the ways that I just mentioned, whether you want to come to a drop-in session or give us a call or send us an email. And we will be answering most of your questions at the Town Square A, I believe, which is very close from here. And thank you for being here. We will be very happy to help you in any way along the way so you can apply for either one of these programs. And I will give you back to Erin. Thank you and good night.
All right, thank you to all the speakers. Hopefully they've answered some of your questions that you may have at the moment. We do have, I think, we might be scheduled until 8:30 in the room, so there's lots of time that if you have additional questions, or we're just in the room right behind. I'm getting head nod, maybe 8 o'clock? 8 o'clock, okay, we're here till 8 o'clock to answer questions. There may be some people online who have also answered questions, so we'll try and get to those right after the presentation here. As I mentioned, I'd like you to follow your passion. Hopefully we've inspired some of that today. If by chance you're still not sure if these are one of your program areas you're interested in, I just wanted to highlight 2 events that are coming up. So in 2 weeks, on a Wednesday, I think in the same room, we're doing an info session for occupational health and safety, food technology, and environmental health, which are public health inspectors. So these are also lab-orientated type programs, but you would be working in a food company or you might be working in a forestry company. So these are more varied types of programs. If you are interested in other health programs or even maybe computing programs or business programs, on February 20th, we have our Big Info Night. And so Big Info Night is where every single program here at BCIT has a table, and you can go from table to table and ask questions about all the programs here offered at BCIT. So it's a great night to come to if you have no idea what you're interested in. But hopefully we've inspired you, and as I mentioned, we'll be in the room next door to answer all your questions. So thank you very much for coming out, and we look forward to meeting with you next door. Have a great night.